Hello, my name is Dr. Rodriguez. I am a science professor. STEAM is an educational movement that combines science, technology, engineering, art, and mathematics. On the iSTEAM channel, we will introduce STEAM fields as access points for experiential learning, inquiry, and critical thinking. Today's episode:
DNA extraction. In this laboratory exercise, you will separate and analyze the components of a plant cell by performing DNA extraction. The laboratory tasks will involve smashing of plant cells, producing a homogeneous mixture of cellular components, and extracting DNA molecules from the nucleus of a ripe banana. Extracting DNA from ripe bananas. Materials: sodium chloride, NaCl, solution. Funnel. Woolite detergent. Meat tenderizer. 2 250 milliliters beakers. 3 10 milliliters graduated cylinders. 100 milliliters graduated cylinder. 1 glass rod. 1 cutting board. 1 knife. 1 electronic balance. 1 blender. 1 hot water bath maintained at 55 degrees Celsius. 1 ice bath. 250 milliliters Erlenmeyer flask. 1 test tube. 4 filter papers. Procedure. Directions to prepare the DNA extraction solution. Use the 100 milliliters graduated cylinder to measure and pour 100 milliliters of 2% sodium chloride, NaCl, solution into a clean 250 milliliters beaker. Add 4 grams of meat tenderizer to the solution. Note: the meat tenderizer will denature proteins. Use the 10 milliliters graduated cylinder to measure and add 10 milliliters of Woolite detergent to the mixture. Use the glass rod to stir the contents of the beaker gently. Note: Woolite detergents will break down cell walls and membranes. Peel a ripe banana and place it on the cutting board. Carefully use the knife to cut the banana into chunks. Each banana is to be shared among 4 students. Use the electronic balance to weigh 30 grams of banana chunks. Place the 30 grams of banana and the previously prepared extraction fluid into the blender and blend them for 1 minute to break open the cells and release the contents. Pour the blended mixture into the 250 milliliters beaker. Place the beaker of banana and DNA extraction solution into the hot water bath that is maintained at 55 degrees Celsius. Use the glass rod to stir the mixture occasionally and allow it to incubate for 15 minutes. Heat will help to denature the proteins and separate the DNA strands. Prepare an ice bath by filling an 800 milliliters beaker with ice. After 15 minutes in the hot water bath, remove the beaker from the hot water bath and place it securely in the ice bath for 5 minutes. Note: you need to push the 250 milliliters beaker down into the ice. Use the glass rod to stir the mixture occasionally and allow it to cool for 5 minutes. Place a funnel over the clean 250 milliliters Erlenmeyer flask and put a folded sheet of filter paper into the funnel. Note: the folded filter paper must be placed in the funnel with 3 layers of paper on one side and 1 layer on the other. Use a little water to moisten the filter paper in the funnel. Remove the beaker from the ice bath and filter the cooled mixture by pouring it on the filter paper in the funnel. When the filtration process is complete, remove the funnel from the flask and discard the filter paper with solid residue in the garbage bin. Swirl the filtrate in the flask, then use the 10 milliliters graduated cylinder to measure and pour 5 milliliters of the filtrate into a test tube. Precipitating DNA Materials: ice-cold ethanol. 5 milliliters freshly prepared banana filtrate. 10 milliliters graduated cylinder. Wooden applicator stick. 1 test tube rack. DNA model. Procedure: use a clean 10 milliliters graduated cylinder to measure and gently pour 10 milliliters ice-cold ethanol on top of the 5 milliliters portion of banana filtrate. Note: the test tube of filtrate must be held at an angle and the ethanol must be poured slowly down the side of the tube. Place the test tube in a test tube rack and observe the contents, especially in the region where the ethanol and filtrate layers touch each other. A white precipitate of DNA that appears as slimy, white mucus should form in the alcohol layer. Record your observations. Move the wooden applicator stick in the DNA precipitate and use it to remove some of the DNA from the test tube. Note, the DNA should wind around the stick. Observe your sample of DNA and compare it to the DNA model provided. Record your comparison.
I hope this presentation was insightful. Please subscribe and give us a thumbs up if you enjoy learning about STEAM. Please also share this channel with someone you think can benefit from or will enjoy the presentation of the iSTEAM channel. Hope to see you soon!